transmission pattern. An individual inherits 2 copies of every gene, except those carried on sex chromosomes, 1 copy from each parent. The copies of a particular gene are called alleles. The specific pair of alleles at a locus constitutes the genotype, and the resulting physical trait is the phenotype. Each human cell contains 46 chromosomes, including 22 pairs of non-sex chromosomes and 2 sex chromosomes, XX in females, XY in males. Gene mutation. Alterations in the DNA coding sequence are referred to as mutations. Mutations can lead to complete failure to express a gene, abnormal function of the gene's protein products. Modifications can be classified either by the nature of the change in the DNA coding sequence or by impacting gene expression change. DNA sequence changes consist of deletions, insertions, rearrangements. Deletions can stretch over large regions, encompassing an entire gene or even group of genes, or can be as small as a single base. Insertions likewise can consist of large stretches of DNA, typically several hundred base pairs, sometimes more, or can be as small as 1 base. Other rearrangements include inversions or translocations. Amino acids, structure and function. It is helpful to classify amino acids based on their R groups because of these side chains. Thus, amino acids with chemically similar side groups can be expected to function in similar ways, for example, during protein folding. Nonpolar amino acids, alanine is one of the most abundant amino acids found in proteins. A D form of the amino acid is also found in bacterial cell walls. Alanine is non-essential, being readily synthesized from pyruvate. It is coded for by GCU, GCC, GCA, and GCG. Proline is the only amino acid found in proteins with an R group that joins with its alpha amino group, making a secondary amine and a ring. Proline is a non-essential amino acid coded by CCU, CCC, CCA, and CCG. Carboxyl amino acids. Aspartic acid, antisocial personality disorder, is a non-essential amino acid with a carboxyl group in its R group. It is readily produced by transamination of oxaloacetate. With a pK of 3.9, aspartic acid side chain is negatively charged at physiological pH. Aspartic acid is specified in the genetic code by the codons GAU and GAC. Glutamic acid, which GA and GAG code, is a non-essential amino acid. It is readily made by transamination of alpha-ketoglutarate. It is a neurotransmitter and has an R group with a carboxyl group that readily ionizes, pK equals 4.1 at physiological pH. Aromatic amino acids. Phenylalanine is a nonpolar, essential amino acid coded by UUU and UUC. It is a metabolic precursor of tyrosine. The inability to metabolize phenylalanine arises from the genetic disorder known as phenylketonuria. Phenylalanine is a component of the artificial aspartame sweetener. In animals and humans, aromatic amino acids serve as precursors for the synthesis of many biologically and neurologically active compounds that are essential for maintaining normal biological functions. Protein, structure and function. Proteins are the workhorses of the cell. Virtually everything that goes on inside of cells happens as a result of the actions of proteins. Among other things, protein enzymes catalyze the vast majority of cellular reactions, mediate signaling, give structure both to cells and to multicellular organisms, and exert control over genes expression. Life would not exist if there were no proteins. Proteins are formed by joining together amino acids, and each protein has a distinct and special amino acid sequence. The significance of the unique sequence or order of amino acids, known as the protein's primary structure, dictates the 3D conformation the folded protein will have. This conformation will determine the function of the protein. Protein synthesis. Synthesis of proteins occurs in the ribosomes and proceeds by joining the first amino acid's carboxyl terminus to the next one's amino terminus. The end of the protein with the free alpha amino group is referred to as the amino terminus or N-terminus. The other end is called the carboxyl terminus or C-terminus since it contains the only free alpha carboxyl group. All of the other alpha amino groups and alpha carboxyl groups are tied up in forming peptides.